My name is Alondra.
And our project is on from DNA to proteins.
Genes provide the instructions for making proteins in the form of DNA. Getting from DNA to protein requires two main stages, transcription and translation. These two processes occur in all organisms. In bacteria, these two processes occur in the same place, but in all other types of cells, they occur in separate chambers. Since bacteria do not have a nuclei, their DNA is not segregated from ribosomes and other protein synthesizing equipment. The lack of segregation allows the translations of mRNA to begin while it's in transcription. In a eukaryotic cell, by contrast, the nuclear envelope separates transcription from translation in space and time, so transcription occurs in the nucleus while translation occurs in the cytoplasm of the cell. This is an overview of DNA within a cell as a way of showing the process of transcription.
So transcription is the synthesis of RNA under the direction of DNA. Since both nucleic acids use the same chemical language, the information is simply transcribed from one molecule to the other. During the process of transcription, we have the template strand and a non-template strand. So for the template strand, our base code is as follows: TAC, TTT, AAA, GCG, and then ATT. Whereas in our non-template strand, we have ATG, AAA, TTT, CGC, and then TAA.
Both nucleic acids use the same chemical language, so the information is simply transcribed, copied from one molecule to the other. DNA can serve as a template for the synthesis of a new complementary strand during DNA replication. In addition to this, it can serve as a template for assembling a complementary sequence of RNA nucleotides. So after the DNA strand separates, the promoter goes through the DNA strands and begins to copy the DNA nucleotides. This is the legend for the DNA template strand and its nucleotides.
So the four bases for the DNA strand are A, T, C, and G. So in this legend, A, which stands for adenine, is for red, thymine is green, cytosine is blue, and guanine is purple. And during the mRNA, the adenine is first thus then transferred to U, which stands for your cell. In this case, our color for your cell is yellow.
As the promoter begins its course through the DNA template strand, it begins to develop a copy of the DNA sequence, making its own DNA sequence, replacing thymine with uracil. As the mRNA continues its trip along the elongated DNA sequence, it begins to expand. The synthesized RNA molecule is called the messenger RNA, mRNA, since it carries a genetic message from the DNA for the protein synthesized machinery of the cell. So the green candy that you see being placed between the two DNA template strands is the mRNA that is getting ready to leave the nucleus to begin translation. As the mRNA is completed, it begins to leave the membrane-bound nucleus, represented by the plate, and begins to emerge into the cytoplasm. As the mRNA begins translation, it approaches the ribosome. Translation is the synthesis of a polypeptide, which occurs under the direction of the messenger RNA. Since during this stage there is a change in language, the cell must translate the base sequence of an mRNA molecule into the amino acid sequence of a polypeptide. As the mRNA molecule approaches the ribosome begins to match the mRNA sequence with codons. As this occurs, the polypeptides emerge after being matched with the mRNA sequence. Using the template strand and the non-template strand that we get the polypeptides MET, LIS, V, ARG, and STOP. An important aspect of this finished DNA into protein molecule, we can see that there's a stop codon as an indicator that that the DNA sequence has been completed.
A smile, I'll tell you.